Hello, everyone. Good morning, good evening. Thank you so much for joining us for another of our monthly webinar series. We hope you've had a great break. For those of you who had a long Easter break, long Easter weekend. And you are ready to hear Karin Kvale from Guiomar in Germany. So thank you so much for being with us today, Karin.
Thank you.
As always, everyone can log into Slido. So that is sli.do in your browser. And the password to get in, the hashtag is OPW, which stands for Ocean Plastic Webinars. And there you can ask your questions. You can also like previous questions from other participants so that those questions come up to the top. And then I'll ask those ones first. So I will now quickly introduce Karin and then hand it over to her. Biogeochemical modeler by training, Karin got interested in plastics when she realized her kids prefer to fill their pockets with plastic debris rather than stones or shells at the beach. Fortunately, she has an awesome boss who let her take a chance for a few months and write a microplastics model for an Earth system model. Her day job includes biogeochemical parameter optimization and uncertainty quantification, which leaves her feeling very comfortable showing you results that come from making a lot of assumptions. So thanks again very much, Karin, and the floor is yours. You can now share your screen and take it away.
Great, thanks. Okay, here we go. Share, yeah, okay, good. And then I bring my presentation up and hopefully you can see it. Okay, I hope you can hear me well. Yeah, good. Okay, so good morning, everyone, and thanks for logging in today to hear my presentation. I'm going to share with you some of the work I've been doing on understanding the biological microplastic particle sink from a global biogeochemical modeler's perspective. So just first a little about myself. Currently, I'm a contract scientist in the biogeochemical modeling department at Guiomar, which is where I started this work. But from next month, I'll be a permanent scientist at GNS Science in Wellington, New Zealand. So just to let you know that my affiliation will be changing in the next weeks, but my current email address should still work for some time. The work that I'll show you today has been completed largely in-house within the GMR Biogeochemical Modeling department. So there was no external project money involved. This was really just very exploratory and involved the ideas of my colleagues Fee, Angela, Shaka, and of course Andreas, who is our department supervisor. So in the interest of time, I'm going to skip a general introduction to microplastics and instead provide an overview of the model that I use. For fairly obvious reasons, nearly all other modeling of plastics have used Lagrangian particle simulation methods. But my preferred model is the University of Victoria Earth System Climate Model, which is an Eulerian Earth System model. So that means it uses a fixed grid and it tracks concentrations rather than individual particles. It's fairly low resolution, so the whole Earth's surface is represented by 100 grid cells by 100 grid cells. So that's 3.6 by 1.8 degrees. And the ocean only has 19 depth levels, with the surface being 50 meters thick. Also, it has a slab atmosphere, which means it's not a proper dynamical representation of the atmosphere. It's just really a very simple approximation to drive the ocean model. But this makes it very efficient to run. So I can get about 300 years of simulation time in about 15 hours on a single processor. And also it has really good ocean physics compared to other Earth system models. These assets mean that the UVic Earth System Climate Model is actively used for a range of applications, from paleoclimate to uncertainty quantification to carbon cycle dynamics. It's freely available and it's fairly easy to modify, which makes it a fun toy model for testing hypotheses. And it's one that I've spent a lot of time modifying the biogeochemical components of. Okay, so I included a representation of microplastic in the model, but when I say microplastic, I want to be clear that the model does not resolve particle characteristics such as particle size or plastic type. So now I know that characterizing particles is a big part of the current microplastics research efforts, and both size and type can influence particle dynamics in the ocean. But as a model developer, my philosophy is to start with the simplest model that I think might suit my purpose, and then evaluate it and revise it. So this is what I do, okay? So in this first model, plastics are represented in units of particles per cubic meter. The plastic enters the ocean along coasts and major shipping lanes, scaled by year 1990 CO2 emissions in these same locations. So this is just a stock forcing file that comes with the standard UVic 2.9 download. And I use it just because it was convenient. All of the plastic enters the ocean as microplastic. There's no breakdown since size is not resolved. The plastic is entering as free microplastic, so that's the MP in this purple box. That free microplastic moves passively with neutral buoyancy, but some fraction of it is prescribed a rise rate. Once the plastic's in the ocean, it can be aggregated into marine snow, so that's the MPA down here, which represents plastic, so then the marine snow aggregation is calculated off the phytoplankton-derived detritus, which is in this gray box, and calculated off what's in the green box. Okay, so the plastic can also be ingested by zooplankton, that's what's in this pink box, and enter the MPZ partition over here. And that represents plastic encased in fecal pellets. So both of the plastic partitions, which are bound in the organic housing, sink at the rate of the detritus compartments. And as they sink, the plastic is released from the organic housing at the rate of detrital remineralization, which is temperature dependent, and it's re-entering this free microplastic partition. If the plastic bound in organic housings reaches the seafloor, then some fraction is prescribed to be lost from the ocean completely, and some fraction is returned to the free microplastic partition in the bottom grid cell. This model does not explicitly represent the growth of algae on a particle, nor this biofouling, defouling cycle that's thought to happen in the upper 50 or so meters of the ocean. But this process is implicitly built into the model via the temperature-dependent microbial loop, which results in this rapid nutrient recycling in the upper ocean. So just to say, we do get a lot of uptake of microplastic by marine snow relative to what actually gets exported out of the surface ocean. Okay, so conceptually, this model is pretty basic, but there were some really big assumptions that had to be made to get the model to perform, two of which are circled here. So full details of this model and the model structure and the parameter sensitivities can be found in the scientific reports manuscript we published last year and in the supplement of that. And I highly recommend reading that supplement if you're interested in the model parameter space and its effects on microplastic transport. The first assumption is that we applied this monod function, so that's what's in this upper yellow circle, to the uptake of microplastic by marine snow. So where the rate of uptake of microplastic by marine snow is related to the rate of detritus formation multiplied by the fraction of detritus that becomes marine snow. So this monod function causes the uptake rate to saturate at high microplastic concentrations. So above a certain concentration of microplastic, the marine snow simply won't take up any more plastic. We tested a wide range of K values, and this model is very sensitive to this parameter. It's also somewhat sensitive to this aggregation fraction. And again, you can find these results in the scientific report supplement. So to get a number of plastic particles uptake rate, we had to go from organic detritus units, which we converted to a number of marine snow aggregates based on an average grams of carbon per aggregate, which was previously published for diatom-derived marine snow. And this monod function is relating the microplastic uptake rate to a number of marine snow aggregates formed at that moment. Okay, so for zooplankton ingestion, there's this relative grazing selectivity for microplastic, which is prescribed. That's what's in this second yellow circle. So that relative grazing selectivity is used in a Holling-2 function, which was already part of the model for, it was a standard representation of zooplankton ingestion of foods. So to be clear, this relative grazing selectivity parameter is implicitly accounting for, implicitly accounting for accidental ingestion, prey confusion, prey rejection. So all of these factors that would contribute to what the zooplankton, how they actually behave with respect to microplastic uptake. Okay, and that's just, it's a gross simplification in a single parameter value. The model is mildly sensitive to the parameter for resulting particle distributions, but it's very sensitive in terms of the biogeochemical effects. So the ingestion of microplastic is calculated based on the maximum potential grazing rate here of the zooplankton, the amount of zooplankton biomass, the amount of microplastic, which is converted to millimoles of nitrogen equivalent for the purposes of the calculation, and the microplastic grazing selectivity parameter, which is weighted against the other available food sources. Okay, so the grazing or the conversion between the model organic units, that's millimoles of nitrogen, to plastic particles is done by first calculating the equivalent amount of food that organic material represents. So that would be in grams of food. This is done with Redfield ratios of the nutrients in the model. So when I know how many grams of food each millimole of nitrogen represents, I apply a food to microplastic substitution ratio, which was subjected to sensitivity testing in the scientific report supplement. And this gives a number of grams of microplastic that substitute for each gram of food in zooplankton's diet. I then convert this number of grams of microplastic to a number of particles based on a previously published estimate for the average mass of a microplastic particle in the ocean. So then I know how many particles of microplastic are being taken up. Okay, and I assume that all of the grazed microplastic is digested and none of it's getting stuck in the guts. Okay, so the plastics pollution problem really isn't a topic suitable for steady state study like many biogeochemical problems are. So when I show you results, all of them are forced with both climate change, so that is increasing atmospheric CO2 following a business as usual representative concentration pathway 8.5, and increasing rates of microplastic pollution. So, partly I'm including CO2 forcing because I'm curious about future scenarios, but partly I include it because the model demonstrates changes in biological activity since the 1950s due to climate warming, and I wanted to represent that. So this plastic pollution rate is taken from Geyer et al. 2017, which demonstrated an exponential rate of plastics production since the 1950s and extrapolated into the future. So the plot here showed the annual production of plastic from that paper converted to a number of microplastic particles and extended to the year 2100. So what this represents is an absolute maximum amount of plastic pollution. So, I mean, there are estimates that suggest less than 10% of the total production ends up as plastic in the ocean per year. But my impression is that this is fairly uncertain, so I play with the input fraction. Okay. So before I get into more realistic plots, I wanted to show some extreme examples of what biological uptake can do to microplastic distributions. So these figures are binned microplastic concentrations in 3 depth brackets. So 0 to 100 in the top row, then 100 to 500 meters, and then below 500 meters. So all of these models have the same microplastic inventory. That is, there's no losses to the seafloor and there's equal pollution rates. These plots are taken from a simulation at year 2020. So that means 70 years of ramped up plastics pollution plus climate change. The leftmost plot is a model where biological uptake is not included, so that's no bio column. So you can see that microplastic is accumulating the gyres in the upper ocean, and it's only reaching the deep ocean in the North Atlantic and this funny spot in the Southern Indian Ocean. Okay, the next column is a model that uses very efficient marine snow uptake of microplastic and has only weak zooplankton grazing. So you can see the effect of distribution is really striking, considering that this is a log scale. So we get this really strong plastic trapping in the biologically productive regions throughout the water column and in the deep ocean, and then total absence in the surface gyres. So there's this huge microplastic inventory in the Arctic and the Southern Ocean, in the deep Southern Ocean, that you don't get without marine snow uptake. Also, it's held in these eastern boundary upwelling systems, and it fills the deep ocean basins even far from plastic sources. Okay, so the third column uses very efficient zooplankton grazing and very weak marine snow aggregation. So this model tends to homogenize the microplastic distributions, and I think this has something to do with the relative rates of uptake and release versus mixing rates, but honestly, I still don't have a good explanation for this. But what it suggests is that marine snow is a more powerful trap for microplastic. Okay, but anyway, in this scenario, the accumulation occurs in all depth bins and it's really diffuse. Okay, and so then the last column combines a very efficient grazing and marine snow aggregation, and the marine snow influence is largely dominating these distributions, but with some differences. So you see the upwelling zones are not as strongly trapping with plastic, but the major difference, I would say, is in the Southern Ocean, in the surface Southern Ocean. So combining these very efficient uptake parameters, both marine snow and zooplankton, is producing the strong accumulation on the subsurface of the surface Southern Ocean that we don't get with other model configurations. Okay, so this observation of particle trapping by biology can be explored further by calculating particle export efficiencies. So for biogeochemical modelers, we often calculate carbon or nutrient export efficiencies by dividing the carbon or nutrient flux across some depth level by their uptake in the biomass in the depths above. So the left plots here show nitrogen export efficiency for phytoplankton, that's the top left. For zooplankton, that's the bottom panel in the left at 130 meters depth. So nutrient export efficiency generally increases with lower temperatures. That's because the cooler water slows microbial and respiration processes, which allows more nutrients to escape the upper ocean layers. Also in this model, grazing is capped above 20 degrees Celsius, which increases the export efficiency of the gyres because there are fewer nutrient losses to zooplankton growth and excretion relative to primary production occurring there because of this capping. Okay, so in the other plots, I'm showing several iterations of the microplastic model with slightly different parameter values in which I plot not nutrient export efficiency, but microplastic export efficiency. So these simulations are not using extreme uptake parameter values. These are selected from a 700 member perturbed parameter ensemble. And there are 3 of the 14 resulting simulations that produced fairly plausible model outputs. Okay, so the top plots are marine snow-bound microplastic export efficiency, and the bottom plots are zooplankton fecal pellet export efficiency. Again, so water temperature has large control on the surface removal efficiency of biological aggregates for plastic particles because organic housings degrade faster in warmer water. But note also in the tropics, with the relatively low particle export efficiencies due to the warm temperatures, these are the same regions that are showing a strong microplastic trapping potential by marine snow. Okay, so following on from the particle export efficiencies, I'm now showing on the right side the resulting top 100 meter microplastic inventories simulated for the year 2020, in which microplastic is taken up by biology. So it's the same simulation as shown on the last slide. On the left side is the comparison model that does not allow biological removal of microplastic. So again, I chose 3 so as not to overwhelm you, but actually there were 14 model simulations in my perturbed parameter ensemble that produced plausible results. Okay, but so LC is a simulation with the lowest surface microplastic concentrations. It also has the most efficient marine snow uptake parameter combination. HC is the simulation with the highest microplastic surface concentrations, but this is for a combination of reasons, so it's not just inefficient biological uptake. MC is an intermediate example with moderate surface concentrations, but also a strong grazing preference for microplastic. So these simulations also vary other parameters, so it's not a straightforward comparison of relative uptake strengths. But the comparison is meant to give a sense of the range of possible distribution patterns produced by this model. So again, as you can see, the addition of biological uptake to the model strongly influences the accumulation patterns. It moves microplastic away from the surface compared to the model without uptake, so that's no bio here on the far left. Log scale is a bit deceptive, but in these models, we don't see significant accumulation, excuse me, in the high latitudes by the year 2100. But here I'll point out that, excuse me, so that the comparisons with real world data are problematic because the model doesn't resolve size fractions. And anyway, as far as I'm aware, the ocean surveys are mostly not sampling the size of plastic that are most interactive with biology. So my best guess of which of these 3 might be producing the most realistic microplastic concentrations is the MC simulation, just based on its rough match in inventory magnitude with the Erickson et al. 2014 simulations, but I would say this is a very hand-wavy estimate. Okay, so I'm now going to move away from discussing the transport and instead show some results that should be published within a week or 2. So there are biogeochemical consequences that result from zooplankton ingesting microplastic in this model. So shown on the far left is the ratio of grazing rate on organic material divided by the prey biomass, which is meant to give an impression of grazing pressure. This is not an explicit model output. The remaining plots show the difference between the models that include microplastic grazing and no bio, the one that doesn't, at year 2020. So what is shown here are largely negative values, which suggest a reduction in grazing pressure on ecosystems with the inclusion of microplastic ingestion. So the reduction in grazing pressure affects organic export production in the model. Shown in the right panels are, again, model differences excuse me, at year 2020 for the models that contain zooplankton ingestion of microplastic and the one that does not. So that's no bio here. The left panel is giving export production in no bio just as a reference. So in some regions, such as the North Pacific, the Southern Ocean, and the Eastern Tropical Pacific, export production increases, and others, export production decreases. So anomalies in export production are mirrored by anomalies in the water column oxygen, which is shown here for year 2020. So in regions in which export production increases due to zooplankton ingestion of microplastic, oxygen decreases. And in regions in which export production decreases due to zooplankton ingestion of microplastic, oxygen increases. So what's going on? Okay, so how the ocean responds to this reduction in grazing pressure depends on whether the regional system is nutrient limited. So looking up here at panel A first, nutrient limited shown in pink. Where's my arrow? Okay. Or nutrient replete, which is shown in blue. So in our model, zooplankton exert a significant control on primary and export production in both nutrient replete systems, which is shown here in panel B, and a nutrient limited system shown in panel D. So consumption of microplastic by zooplankton in nutrient replete environments, now moving to panel C. This is reducing the grazing pressure on primary producers, and that causes an enhancement of export production. That upon remineralization at depth is, consuming oxygen. So this accelerates oxygen loss, which is also an effect of ocean warming due to climate change. Okay, so now looking at panel D, in nutrient-limited environments, primary producers rely on recycled nutrients, which are supplied via microbial loop as well as zooplankton excretion. But in the presence of zooplankton ingestion of microplastic, now looking at panel E, a greater proportion of nutrients cycles through the temperature-sensitive microbial loop, which leads to a decrease in export production, which in turn drives a reduction in oxygen consumption at depth for mineralization. Okay, so in the nutrient-limited regions, the ingestion of microplastic by zooplankton leaves more oxygen available in the water column, but in nutrient-replete regions, it leaves less. But unfortunately for the ocean, the net effect is one of accelerated oxygen loss. Okay, so at a global scale, this oxygen loss is significant. In our model, the loss of oxygen due to climate change since 1950 is about 1%. So that's shown as the dashed black line here. The colored lines represent the 14 members of the perturbed parameter ensemble that was published as part of the scientific reports manuscript. All of these produce plausible microplastic particle inventories using plausible biological aggregation rates and input fractions. So across the member ensemble, oxygen loss ranges from an additional 0.2 to 0.5% by year 2020. And by year 2100, this range has grown to an additional 0.2 to 0.7% oxygen loss beyond the effects of climate warming. So I want to just restate that this is a model first result. And while its sign is robust across the model ensemble, its magnitude remains sensitive to model parameters. It's particularly sensitive to the zooplankton grazing selectivity parameter, which is again representing the many complex vectors that influence what zooplankton eat. And I want to be clear that so if the zooplankton do not eat the microplastic, this additional oxygen loss doesn't happen. But within bounds, the model is more sensitive to the readiness of zooplankton to eat microplastic than how much microplastic is actually available at the surface. So I can try to link this back to my daughter's loving to pick up plastic bits at the beach. I mean, if they're interested in it and they go for it, then you have an effect. If they're not, then you won't see this effect. Acknowledging that zooplankton aren't it's not a perfect analogy for zooplankton interactions with microplastic. But so, I understand that there's a lot of work that remains to be done with regards to zooplankton and microplastic dynamics, and that this one parameter is really a gross oversimplification of the real world. So I take this result as a strong motivator to improve the model and to learn more, which is why I'm presenting these results to you, because I'm hoping that the broader community can offer their expertise in improving the constraints on this model. Okay, so, but if we say now, okay, maybe it is possible that even at low concentrations, microplastic could have a globally significant influence on biogeochemistry, then this, for me, raises very relevant questions for future surface removal and microplastic pathways and how these might feed back on ecological responses to climate warming. So, note that those nutrient-limited regions, which are shown here in pink, include the major ocean gyres, where no plastics, including microplastics, are accumulating. These nutrient-limited regions are expected to expand with climate change-induced stratification, and as they do, export production is expected to decrease in them as nutrients become more scarce. So a decrease in export production in the stratified ocean could lead to even more rapidly increasing microplastic concentrations at the surface, which could further suppress grazing regionally, which could subsequently reduce export and compound the biogeochemical effects of climate change. Likewise, we see the potential for a microplastic trapping feedback in the nutrient-replete tropical ocean. So unfortunately, I don't have time to go through the analysis here, but anyway, the loss of oxygen in the tropical upwelling zones. Okay, this is a powerful mechanism via the nitrogen cycle to accelerate primary producers with increasingly inefficient export due to climate warming. An acceleration of marine snow formation downstream of upwelling regions due to the loss of oxygen induced by zooplankton eating microplastic encourages further trapping of microplastic in the surface and marine snow and further ingestion of microplastic by zooplankton. So we see this biogeochemical effect in our model, but I'm only now working on quantifying these feedbacks in terms of the magnification of surface microplastic accumulation. So stay tuned. I've posted my email where you can reach me for at least the next few months as I need to Janice. And I'd like to open the floor to questions.
I can clap for everyone who is who's joining virtually. Thank you so much, Karin. That was super interesting, and so many complex pathways, so many different components of the model, and yeah, you've looked at different scenarios, and it's really interesting to see what's happening in these future scenarios with climate change and how that can really affect the biogeochemical cycle. So yeah, thanks so much for this quick overview of two of your papers. So there are some questions that are coming up on Slido. So everyone who's joining, remember that you can go onto, in your browser, you go to sli.do, and then the password to get in is OPW. It's here at the bottom of the slide that you can see. Karin has put kindly on her slides. Okay, so maybe Karin, now you can, well, actually, yeah, I did have one question that maybe you can go back into your slides. So yeah, it was actually the very first one from this second paper, and where you have on the left, every time that you have this no bio scenario, I was wondering what the, yeah, exactly. So here you have LC, HC, MC for low, high, and moderate. And so for the no bio, what are the concentrations there? Is this kind of the, yeah, what concentrations are the microplastics being input?
Being input into,
yes. Mm-hmm.
I would have to look. I'm sorry, I don't remember.
No worries. I was wondering if it was one of the three scenarios, or if it was, because here, for example, the no bio and the HC seem to be very similar.
Right, but this, so they're using comparable. I think for my inputs on the no bio, I'm using kind of an average between these three.
Okay.
Okay, so with this perturbed parameter ensemble method, this is Latin hypercube methodology, where you give MATLAB some range for each of your parameters, and you say, okay, pick some combinations for me, and then you run, 700 combinations. And so all of these models are a little bit different, but the no bio was using kind of an average input rate. So it's somewhere in between all of these. And so the reason why HC would look similar to no bio is it has something to do with the biological interaction. Probably has relatively less biological interaction than the other models.
Yeah. Okay, great, thanks. Okay, so maybe you can stop sharing your screen, and then I'm going to go on to Slido and ask some questions that we have from the participants. Okay, so Riata was asking, does the model consider diel vertical migration of zooplankton that might transfer microplastic from the surface to the deep ocean?
No, it doesn't.
Okay. That's a simple answer.
Go for it. So the code is freely available. Anybody who wants to modify it, try your hand at it. I'm always happy to advise or help in any way.
That could be quite a good, if you can maybe send the link, is it on GitHub or something, then yeah, maybe if someone's interested, they can take a look at the code.
Yes, it's on GitLab here at Geomar, and you have to register, but then once you register, you're in the system, and
Okay, and then they can search your name and it will come up. Okay, great. Well, there you go, Riata, if you want to check it out. And yeah, if you have a follow-up question to that, you can also ask it on Slido. Then we have a question from Lilia, who says, thank you for a great lecture. In the second study, you did not account for marine snow aggregation with microplastics, but only zooplankton grazing, right?
No, so in the second study, I am using aggregation in the marine snow as well, but the only biogeochemical effect in that model is the zooplankton ingestion of plastic.
Okay.
Yeah.
All right. Yeah. Okay. Well, again, Lilia, if you have another question that is kind of yeah, why you were interested in this, then you can also ask it on Slido. Then I had another question that I put on Slido. I have some more on my sheet here in front of me too, though. So you said in the first study that you had this assumption of the microplastic being consumed by zooplankton, so being completely consumed by the zooplankton, and none of it being stuck in the gut. And I was wondering if this was based on some study, or if it was just because you had to limit the parameters in some way.
Yeah, it's just a pragmatic, keeping it simple.
Yeah, and yeah, go ahead. Sorry.
Yeah, this is something that could be explored.
Right. Yeah, it's always hard, with these models to know how much you should do sensitivity analyses, or if you just choose kind of some assumptions and then build your model of that. So did you do any type of sensitivity analyses to see if some of these assumptions could really change the results a lot?
I mean, so I tested plausible parameter ranges in the scientific report supplement, and to look at how this would affect transport. But I haven't done enough, and I mean, I think there's more that needs to be done. I mean, so this monod function, for example, this was the brilliant idea of Phi to just include a monod function, but we used it mostly for numerical stability because linear functions were causing the model to crash. So I think this would be another direction to go with an excellent student project to test different possible parameterizations of microplastic aggregation.
Right. Yeah. There's always more to do, right? There's endless things that we can always do.
But it's super, yeah, and I don't think that, as far as I know, there's no nothing published on this. And what kind of parameterization you pick? Do you pick a monad function, some kind of linear function? I don't know. I mean, it's hard to stay on top of the literature. There's so much coming out every day that it's really hard. I find it very difficult to know what is happening in this field at the moment. But this is important.
Yeah, I guess that's also an interesting point to make, that there is a huge amount that's coming out, and a lot of the time you might be the first to kind of be looking at this particular question from this angle, and so you're trying to stay on top of things, but also trying to kind of bring together the different parts of the literature that could be used, right? So okay, so we do have a follow-up question from Lilia, who says, do you think we can prove some of your results of numerical modeling using the available field data, or which field data do we need to collect to support your results?
I would love it if somebody could go and assess how realistic these results might be. Looking at changes in dissolved oxygen, looking at changes in growth, I think, in trying to relate this to the contamination by microplastic. I mean, this is all useful information.
Yeah, I think, as you know, this is quite related to the work that I'm currently doing, and especially when you're trying to do some global scale models, and really trying to see the effects of these nano microplastic type sizes. It's just so hard to go out and actually measure these processes. So yeah, I think it might be a few more years until we can actually get some kind of global scale field measurements of these kinds of things. Okay, we have an anonymous question that says, can we say that the intensity of microplastic vertical transport deeply depend on POC vertical attenuation expressed frequently by the Martin curve?
Yeah, I've been pondering this because I've started to look at some very long-term simulations, and I don't have a good answer yet. I mean, I think we don't really know enough, right? And there's a lot of sort of microscale processes that aren't represented in this model that could be important, and different particle types, and the particle dynamics. I mean, they don't really resolve this, so I can't say for sure. But what we need is better data, I think, with respect to depth measurements. So one thing that I've seen in this model is that the particle profiles take on very distinct profiles depending on the parameter combination. So you can have, very, very high concentrations at the surface that go very low and then they get much bigger, or you could have, just a very gradual change in the particles with depth, and so these different profiling, really getting a better handle on characteristics of the profiles in the real ocean could probably inform better on the major processes that are driving the particle distributions. Yeah.
Great. So now we have a question from Heinz. So I wonder if you could tell us some more about the sensitivity to remineralization rates and if there are other loss processes of microplastics of the MPZ and MPA.
Okay, so I guess in the scientific reports supplement, I didn't go into remineralization rates.
Okay.
I have done some testing of the remineralization rates, and I'm looking through these results now. I mean, so you don't see a major change in the distributions of particles. You know, on the at global scale, they're still basically going to the same places, regardless of, you know, within bounds of how you adjust your remineralization rates.
Mm-hmm.
But this can affect, say, the oxygen sensitivity.
Right.
So biogeochemical response is sensitive.
Okay. And I guess, yeah, if the particle can then, you know, resurface, or, you know, the vertical distribution of the particle, depending on this remineralization rate, right? So yeah, the loss of its organic housing. I quite liked that that term that you used. Yeah. Okay, great. And yeah, and Heinz also said very interesting and numerous results. So that's a nice comment to add too. Okay, so let me just check. So Riata has another question, and this one focuses on so the production rate of larvaceans can be as high as copepods, and they efficiently trap microplastic with their house, which might be interesting.
Okay.
So I'm not sure if it's probably more of a comment, or is this something that you've looked into, or that
so this model only has one zooplankton that's responsible, representing all the zooplankton of the world. I mean, but there are other directions you could go with it, and there's certainly a lot that can be explored with the model. This is just a first attempt.
Okay. Yeah, I see that. Yeah, it's a very kind of high level, you know, model where, as you mentioned at the beginning, everything is quite like coarse, and it's really just to start getting the first look on what these processes, how these processes can affect microplastic distribution, and so yeah, it kind of makes sense that the zooplankton is also quite it's not looking at specific species. Okay, let me take a look. Okay, well, Riata had a kind of like a next question that kind of follows up on that, but I guess that all depends on the model that you're using. But do you have a plan to include the contribution by larvaceans? And he specifies appendicularians.
I do not have this as one of my plans. Who is enthusiastic about these sorts of additions, by all means, email me. I can offer advice from a distance. I mean, so, you know, in my new position, and now plastics is not my primary research topic. So this is something that I so far can be done on the side, and it will probably have to stay something on the side at least until I get some kind of funding for it. So.
Yeah, well, it's incredible that you've got two, you know, these papers out that are, if this is what you're doing on the side, that's amazing. Okay, yeah. So I guess these kind of questions that are a bit more specific about the biological species or the components, you know, this is something that once the model is kind of running, then, you know, depending on the literature parameterizations, you know, you can kind of change these rates of grazing or whatever it is that, you know, can affect the microplastic consumption and and that whole cycle. So, okay, I believe that's all the questions on Slido. Now I have a few other questions. So this oxygen loss that you were mentioning, this 0.2 to 0.7% that could be affected by 2100. So I was just wondering if, you know, you say that already 1% of the oxygen has been lost in the last, I think it was 100 years or more. And And so I was kind of wondering what the kind of the overall effects of this, you know, if there are some estimates of how this can impact, you know, at what extent this would actually impact the whole ecosystem. You know, I know that obviously oxygen depletion is a big problem, but you know, at what kind of you know, what range is kind of this dangerous range, I guess. Are there these estimates?
Yeah, well, so it depends on where you're losing oxygen, right? So I don't think probably anybody cares if oxygen disappears from the Southern Ocean, at least not for a while, because there's already a lot of oxygen in the Southern Ocean. And the places where oxygen loss is really going to matter is the places that have already very low oxygen concentrations that may be near a, you know, anoxic threshold that would be relevant for the biology. So, you know, so the Eastern Tropical Pacific, this would be a region where I'd be very concerned about the potential for microplastic to be accelerating oxygen loss.
Yeah. Yeah, that makes sense. It's really kind of these two different zones that you were mentioning in the pink and the blue at the end. Yeah, that it's a really, you can't really say that there's this kind of global effect of this, right? And it really just depends on the zones. There is a response, I think, to one of Riata's questions, and that's explaining it a little bit more of what you were mentioning. So unfortunately, there are very few global models that include larvaceans despite their high production rates, and also other zoo groups. More work to do. Yeah, it's true that it's quite hard to, I guess, to really model, you know, there are just so many species, and you know, there are different species that are important in different regions too. So I think it must be a really hard job trying to decide, you know, what is the most dominant species that should really be that should be modeled. So it's true that these modelers have a lot more work to do. Yeah, and I did have one other question. So I was wondering if you started looking at other trophic levels. So I know that you've looked at zooplankton grazing, but are there higher trophic levels in this model that you're using? You know, can there be, you know, this other level of grazing, of consumption of the microplastic by larger species?
So far, no. We have one of my colleagues here, Julia Getzlaff, is writing an approximation of mesopelagic fisheries. So this might be something that in the future could be coupled to explore how you know, the microplastics that's settling in the deep ocean might be influencing, you know, higher trophic levels there. But writing a fish model, as far as I'm aware, this is a considerable amount of work that would probably require some years for even an experienced model developer. So this isn't something that I had planned, but again, if someone wants to take it on, I'm happy to advise.
Yeah, for sure. Yeah, okay. I believe that's all the questions here. If anyone else has some more questions, there are still a few minutes. And otherwise, I would like to see if Karin, you have kind of some concluding thoughts and, you know, some ideas. So the whole point of these webinars is to try to see, you know, if there are people that can maybe you know, get connected through this network. And so if there is, you know, one kind of main missing piece that you would like to be working on, and you know, people that you would like to connect with for your research, then, you know, if there's kind of one call to experts, who do you think would really benefit, or how do you think you would benefit from these kind of collaborations for your future work?
Okay, well, so I would really love to hear from people who are characterizing microplastic down the water column. I mean, this is at the sort of biologically interactive level. This would be crucial for reducing the uncertainty in this model.
Mhm.
And also anybody who is working with particle aggregation dynamics, I think this would also be very, very useful for constraining this model. It needs more constraints on it. I could just run with it, but I think that maybe that's not so responsible. I don't know. You can make lots of pretty pictures, but if you have garbage coming in, it will be garbage coming out again too. So we need more data to make this model, have more confidence in the model.
Yeah, and it kind of made me think of one last question. So the biofouling components that you mentioned is not explicitly modeled. Is this something that you're planning to kind of develop further, or is it in your plans?
No, it's not, because the surface is 50 meters thick.
Of course.
So this is something that's better left, I think, to Lagrangian approaches like your own. But it might be useful, and I have to say now, I haven't read your paper completely yet, but I think I wonder if maybe some of your results might help to improve my model.
And maybe, yeah, maybe even the work that we're doing right now, because that's going to be even more kind of focused on on some upper ocean mixing, and so hopefully we'll have even more of the physical components that can affect the particle distribution in the vertical. So yeah, let's see if we can work together in the future. Yeah. Okay, great. Yeah, I believe that's all the questions on Slido, and yeah, so I would just like to thank you again, Karin, for your time today and for sharing your research, and for everyone else who's joined, please stay tuned. We'll send out the email for the next the next webinar next month. That will probably be on the first Tuesday of the month, like we always do at the same time. Unless we state otherwise. So thank you very much, everyone, and have a great day.
Thanks for having me. That was fun.
Yeah.